freezing in here this morning. So, there we go. But remember, this is a diagram that shows the reasons, the 3 reasons why a neuron has a resting membrane potential of -70. So we're going to start, we're going to look at several different potentials. So yesterday we looked at 4 different types of channels. We looked at leak channels and ligand-gated channels, voltage-gated channels, and briefly at mechanically gated channels. All right? And those were just protein donuts, if you will, that allowed ions to pass through the cell membrane. Now, when ions pass through the cell membrane, they do so because when they do so, a current is created. And the ability to create that current, or the current itself, is identified as a potential. So our first potential that we finished discussion with yesterday was resting membrane potential. And if we diagram, and what we're looking at is on the cell membrane or on the axon. Remember, it's just across the membrane. There are more positive cells along the outside of the membrane and less positive or more negative cells along the inside. And the difference in the number of positive and negative ions is what we call the resting membrane potential. So we look at the graph, or the chart here. Our resting membrane potential is about -70. Some graphs will show this at a -90. It depends on what neuron, what species we're looking at. So that means that there's a sufficient difference in the number of charges inside and outside the cell that if they were to come to equilibrium, if enough positive ions would flow into the cell to bring this to 0 difference, it would create a current of 70 millivolts. All right, enough ions would have to flow into the cell that would create a current of 70 millivolts. So, before we move farther, based on the diagram that I have on the screen, what are the 3 conditions within a neuron that creates this -70 millivolt resting membrane potential?
Depolarization, repolarization, and hyperpolarization.
No. Negatively charged proteins. You're talking about what we call the changes to the resting membrane potential. Okay? So negatively charged proteins create a negative charge inside the cell. That would be these structures here. Okay?
Greater number of leak channels.
Okay. Which one?
Potassium.
More potassium leak channels, which causes us to lose positive ions. All right? And then the sodium-potassium pump, the flip-flop.
With the 3-to-2 ratio.
Correct. And then the sodium-potassium pump with a loss of positive ions. So that creates our resting membrane potential. That's what we have to start off with. Because if we have no resting membrane potential, then if we open a voltage-gated channel or we open a ligand-gated channel, nothing's going to happen. All right? So I need to have, for instance, a lower pressure, fewer molecules per cubic millimeter or cubic centimeter, if you wish, for air to move into my lungs. If I just open my mouth, there's no airflow in and out. I have to push air in or push air out. Okay? So there has to be some concentration difference for ions to move into or out of the cell. And that's why we have a resting membrane potential. So how do we can raise and lower that resting membrane potential without opening or closing or doing anything with our channels yet, by just changing the environment. So you have this table on page 10. So when we talk about decreasing the resting membrane potential, what we're saying is we're moving it farther away from zero. So if I change the resting membrane potential from here to here to a minus 95, all right, what is that saying about the inside of the cell versus the outside of the cell? It's more negative, all right? Now, I can also do that. I can make the inside of the cell more negative, or I can make the outside of the cell more positive. Okay? So, with potassium ion concentration, let's say that I have a cell membrane here. This is outside the cell. This is inside the cell. And here are my normal potassium leak channels. And I normally have more potassium inside the cell than I have outside the cell. Correct? So my normal diffusion pattern is to lose potassium ions. This would be the normal situation. So this is normal. Now let's take the first event here, and we're going to increase the number of potassium ions outside the cell. We're going to give this person an IV, and we're going to try to murder them without anybody knowing what's happening. So we're going to give them an IV with a high number of potassium ions. And we're going to increase the number of potassium ions outside the cell. What is that going to do to the normal diffusion of potassium through our leak channel? It's going to stop it from moving out, and if we make this really high, it might even add some. Does that make sense? Because we normally go from a high to low, and if it's normally this gradient and we make it this gradient, it's not going to move as rapidly. Remember our discussion on simple diffusion? Okay? So that's going to make the inside of the cell more positive. So it's going to move our resting membrane potential higher. Okay? So it's going to decrease the difference between the outside of the cell and the inside of the cell. Does that make sense? I don't want to go so fast that you guys are just writing it down and not following along. So it decreases the difference, in essence makes the inside more positive because they're not moving as fast. Not just because of the concentration gradient difference, but because there's a change in the electrical force. Remember what we meant by electrical force? The positive ions are attracted to negative ions and repulsed by positive. So the more positive I put out here, the more resistance they're going to be for more positive joining them. Okay? So that's going to raise my resting membrane potential. So this is going to be what happens with increased extracellular potassium ions. Now, there is a term called a threshold potential that I'm going to talk about in a few minutes. The threshold potential is the voltage required to open our voltage-gated channels. Remember that picture I showed you yesterday about with the paddles? And so we had maybe that was forward. We had these voltage-gated. Come on. Go back far enough here. Everybody have this image? And it took a certain current of ions flowing by to swing our paddles and open the channel. Well, if the inside of our cell is already has more positive ions, we're halfway to our goal, maybe, and so the current going by only needs to be half as strong because we already have a bunch of positive ions in there. Does that make sense? So let's say that I need to bring the current on the inside of my cell to a minus 30 or a minus 40, just to pull a number out of the hat, typically it's around a minus 50, for those voltage-gated channels to open. If I keep my potassium ions from leaving, that might push me up here to a minus 60. Now my incoming current doesn't have to be as strong because I'm already partway there. So what this does is it makes my cell more irritable. It makes it easier for it to open the voltage-gated channels because it doesn't take as much of a trigger to get those voltage-gated channels to open. All right? So we're back to the dominoes, and the domino pushing over is the voltage-gated channel. All right? If I put that domino on a little bit of, a teeter-totter kind of thing, where it's unstable, then it's not going to take as much force to push it over. So that's what we're doing by changing where our resting membrane potential is. Now, let's go back to this table. If I do the opposite, and I remove potassium ions, okay, I give the person a drug that soaks up the potassium ions and removes them from the body, then what's going to happen to the rate at which the ions leave the cell? It's going to leave faster because what I've done is tilted that gradient, and therefore these are going to move out much more rapidly because there's fewer positive charges out here to repel them, plus my concentration gradient is steeper. So when that occurs, which way is my resting membrane potential going to move? Down. Okay? So it's going to move farther away from my threshold. And now it's going to be more difficult to get my voltage-gated channels to open. They still require the same current, but I'm going to have to add more ions. I'm going to have a stronger stimulus. More positive ions are going to have to enter the cell to get to that current. And so it's going to be less likely to fire. It's going to inhibit. I'm more likely to have a slow heart rate, be unconscious, because my neurons are sleepy. And that's it. They're not really sleepy, but they're not going to be as sensitive. Okay? The second event has the same effect as this first one here. By how would I increase potassium ion permeability? More leak channels, exactly. So if I add more leak channels, and this would happen because the neuron cells genes are active in making more leak channels. Right? So if I can turn on those genes and add more leak channels, then I'm going to increase the availability. I'm going to lose more ions, and that's going to give me the same result here. The inside of the cell is going to become more negative. So this is also going to occur with an increase in the number of potassium leak channels. All right? Now, this is physiology. Okay? This is the difference between anatomy and physiology. Anatomy is more about structure. So anatomy would be understanding the structure of the neuron. How does the plasma membrane, even the structure of the plasma membrane, the structure of those channels kind of overlaps between physiology and anatomy. Physiology is function. What are these ions doing? How do those channels change shape? People ask about this time of the semester, is 431 going to be harder than 430 because I'm barely making it through 430? And my standard answer is, it depends. There's still anatomy in 430, but there's more of the physiology. A lot of students coming from high schools have only learned how to memorize. And they know how to do that really well. But it's a struggle to figure out how something works. So if that's the case, then 431 might be a little bit harder because we still have the anatomy, but there's more of this figuring out how something works. We're going to repeat the same information when we do the heart. That's why I said this stuff is really important for next semester. So we're going to go over the same information when we get to the heart, and if you learn it now, that's going to be just a real quick review. But some people find this more fun. Once you understand what happens, it becomes logical sense. And it's not a memorization of a bunch of nonsense phone numbers that you have no way of recalling unless you just go over it every day. Because there's nothing that brings it to mind. Whereas in this, one thing has to happen before the next, and rather than memorizing word for word each step, you understand the process. And once you understand the process, each step reminds you of the next step because it's a logical sequence. Okay? And in fact, if you're a visual learner, which I am more than other types, I actually see this as a little movie. And I just play the movie instead of going through. Step one is, what are the words for step one? You don't memorize each word, you see the event. Okay? And so animations of these events online or through my AMP, if you have access to that through a new textbook or you purchased it, can make that visualization easier to understand. Okay? All right, so sodium ions. So I don't want you memorize, what I'm trying to say in that back-around way is I don't want you memorizing this chart. You can figure it out by drawing these little pictures. So if it doesn't come to you right away, make a little diagram. If I had a whole bunch of sodium ions or positive ions outside the cell or inside the cell, what's that going to do to my resting membrane potential? Okay? So it's a cause and effect all the way through. All right, so obviously if we increase sodium permeability, if we add sodium channels here, which way is sodium going to move? Into the cell. So what are we doing to the number of positive ions inside the cell? Increasing it. So we're doing the same thing here when we increase potassium ions. We're raising moving our resting membrane potential closer to the threshold, making it easier for the cell to open voltage-gated channels. Now, to show that faculty don't always see things that they should, all right, and how we get into ruts, for years, when I thought of calcium ions in neurons and muscles, it was in the concept of a muscle contraction. And as we'll see in our next unit, calcium ions bind to proteins in our muscle cells and unlock the access between actin and myosin. We talked about actin and myosin. When actin and myosin slide past each other, look at the distance between my elbows. When they slide past each other, the myosin pulls on the actin. The distance between my elbows is shorter. That's how a muscle cell contracts. Well, there are proteins that block actin and myosin from interacting with each other. Calcium allows those proteins to move. Okay, that's the way, for years, I understood calcium. And it's true. That's what calcium does. It's not like I had a misunderstanding. So I remember my friend that broke both his scapula and his clavicle in a bicycle accident. Okay, so the same guy rides about 50 miles a day, 100 to 200 miles on the weekends, and he gets muscle cramps. As does my husband, who only rides about 60 miles on the weekends. And he said he carries Tums with him and takes them during his ride. The increased calcium blocks the cramps. So when he gets home or while he's riding, his legs don't seize up into cramps. So he asked me to explain the physiology of this. So I started off with calcium and muscle contractions, and I stopped midway through and I go, wait a minute, that would mean all that calcium you're taking would make you more likely for your muscles to contract and go into a spasm. And then I got blocked and stumped. It's like, this doesn't make any sense. A week or two later, I was giving this lecture, which I had given multiple times before, and I got to this chart, and I read this out to my students, and all of a sudden it was an aha moment. Stupid me, duh. This is why calcium stopped him from having cramps. Calcium indirectly blocks our sodium voltage-gated channels from opening. So a nerve signal, when you're tired, or you get little twitches, okay? So nerves can depolarize on their own when you're tired and we used up ATP and we aren't using our pumps to put us back into homeostasis. And so he was getting these spasms, nerve axons firing and causing his muscle to contract because his pH was all out of balance and so on. When he took the extra calcium, it bound to the sodium channels and blocked them from opening and stopped the nerve from sending signals to the muscle cells. It's like, that's how it works. And even though I've been saying that for several different lecture periods, it wasn't illustrated until I had an actual hands-on case of how it would block the muscle contraction. So when we talk about voltage-gated channels, it's, if we look at our sodium channel, there's a little site for calcium, and when calcium is there, it keeps the sodium channel from changing its shape. So when it can't do that, obviously ions can't move into the cell, and it can't respond to a signal. So if we have extra calcium in our body in the extracellular fluid, we have decreased response, but we haven't really lowered the resting membrane potential so much as we prevented the channel from responding to a signal. Okay? And that's, of course, if we have decreased calcium, then it's more likely to respond to just slight change. So all of this was resting membrane potential. The normal resting membrane potential, and then making it closer or farther away from the amount of ions needed to trigger a threshold. So that brings us to the term graded potential. We've been talking about this obliquely for a couple of weeks, because I introduced you to the concept of graded potential the first time we talked about a ligand-gated channel. Now, I don't eat meat, but meat is graded, I know that much. Okay? So the ag department grades it, grade A. They never seem to sell grade C meat. I don't know if they just feed that to other animals or something. But meats always seem to be graded A, or at least that's the only time they advertise it. And so we get grades in classes, all right? And so if you have a high number of points on exam or a low number of points on exam, it's graded. So graded means an incline can be graded, a 7% climb or a 10% incline. So that means that there's different levels, okay, when we talk about a grade. When we talk about a graded potential, we're talking about the fact that the number of positive or negative ions that enter a cell can be different. Well, that has to do with ligand-gated channels. If I open one ligand-gated channel, I get a certain number of ions that flow into the cell. If I open 500, guess what happens to the number of ions in my cell? It's going to be different. So a graded potential is one that can be a different value. It can be a low value, it can be a high value. Okay? A potential can make the inside of a cell more positive, or can it make the inside of a cell more negative? So graded potentials are always formed by ligand-gated channels. Am I writing this properly? So our resting membrane potential was associated with leak channels and sodium-potassium pumps. Our graded potentials are associated with ligand or chemically gated channels. That's how we create that. Our action potentials are associated with voltage-gated channels. When a voltage-gated channel opens, the same size of current, the same charge of current is created every time. One neuron may create a certain amount of current, but it will be that current every time. A different neuron may have a different current for an action potential. But graded potentials vary depending on the number of channels that open and on the ion that enters into that channel. All right? Another thing about ligand-gated channels and graded potentials is that there's no way of perpetuating them. These currents will eventually dissipate, peter out. Okay? So they may start out weak and get even weaker. They may start out strong and get weaker. They never start out weak and get stronger. And they aren't perpetuated. We don't create them over and over. So they have diminishing strength. Or current value. So where would we create a graded potential? At the synapse. So what we've been drawing, and we're going to actually finish up lecture today by talking about a synapse. But if we have our synaptic bulb, and we have a dendrite, I'm just going to simplify this and create just a few dendrites on our cell body. All right, so it's the cell body. And the dendrite where we form graded potentials, because those are formed by ligand-gated channels. So the reason I have the picture of the water, the rain falling on the water here, is because, as I talked about earlier, the ripples created by that are going to get less and less energy as that ripple moves farther away from the site. We can create a big ripple by dropping a big rock in the lake, all right? We can create a small ripple by dropping a small rock. Now, if I create a big ripple, a wave, by dropping a big rock, that's going to go farther across the lake. It's going to last longer. It's going to have more energy than if I did a small one. Okay. So we have the neurotransmitter, our ligand, binding to the channel. And this is an example of a direct ion channel where the channel is the protein that binds to the neurotransmitter. So on Tuesday, we're going to start looking at the different types of receptors. But when that opens, it's going to allow the ions to come in. We'll talk on Tuesday as well about what happens to that neurotransmitter and the fact that it's being broken. So this is a local current that's being produced. Let's draw kind of a larger view of what's happening right here. So here's my synaptic bouton. Inside, I have synaptic vesicles with neurotransmitters. And let's stick with a common theme. We'll still say this is acetylcholine. It's one of the most common ones. And then we'll draw a larger picture of our dendrite. And so when this neurotransmitter diffuses across the space, or synaptic cleft, the synapse is the relationship between these two neurons. Okay? So we will have a receptor that opens when it binds to the neurotransmitter. Once that opens, let's say 10 sodium ions come into the cell. All right? If we look at that on our graph, it's going to change our resting membrane potential. And it's going to just make a small little blip. going to go up a little bit, and then as the ions diffuse through the cell and out the cell, it's going to come back down again. Okay? Not enough to really do a whole lot. Let me move this other diagram down here just a little bit. So those positive ions coming into the cell are going to just come in little ways, and then they're going to get pumped out of the cell. So they're not going to have a current that's very strong. It's certainly not going to diffuse all the way through the cell to the axon. Well, let's say then that I open up 10 more of these channels. And each of them allows, and I'm not counting anymore, each of them allows 10 ions to come in. Now I've increased my potential. So now I would see something that looks like this. And these ions coming in are going to move a little bit farther into the cell. But by the time they reach my axon, the current isn't strong enough to open up my voltage-gated channels. Okay. So instead, what I do is maybe I make this one fire 10 times in very quick succession. All right, it's going to fire and fire and fire and fire. It's going to release more and more and more neurotransmitters, and these channels are going to stay open for a longer period of time, or if they close, they're going to reopen right away.
So is the acetylcholine also the ion? Or
No,
is there...
acetylcholine is the neurotransmitter, and the pluses are the ion.
So the ions are just kind of floating in the...
Yeah, they're in the extracellular fluid.
All right.
Okay. So now if I cause this to fire multiple times in a very quick succession, I'm going to get a whole lot more ions, which is going to create a stronger current, all right, as it flows. Again, some will go out here, some will go out here, some will leave, but I'll increase the amount of ions that actually reach my axon where my voltage-gated channels are. So that is known as summation. Instead of, I could have one neuron firing, 10 times really fast succession, right here. Or maybe I could have 10 other neurons doing the same thing. And they are all firing, by firing I mean setting an action potential and releasing neurotransmitters. And all those neurotransmitters are opening ion channels. Okay, all working at the same time. And so that's going to increase the flood, and it would take my membrane potential and make it even higher. Again, that's what we mean by a graded potential. I can have a weak one, or if I change the situation, I can have a strong one. Okay, yes?
What did you say that was called again? I guess we're...
Summation?
So is that increasing the firing, or...
It depends on the effect. This is talked about on the bottom of page 12, summation. It depends. It would increase the release of neurotransmitters from, so with multiple firing, I have an increase in the release of neurotransmitters. Whether it causes this neuron, the postsynaptic neuron, to actually fire depends on what those neurotransmitters, what kind of channels those neurotransmitters are. In the example I've given here, they all open positive ion channels. So I increase the number of positive ions. But let's change it. Let's say that one of these that opened here, instead of opening a sodium channel, opened a potassium channel. And this one opened a potassium channel. So instead of adding positive ions, I'm going to start losing positive ions. So now the number of ions that reach my axon in this summation is kind of like if I add 10 but take away 2, I end up with 8. All right? If I add 10 but take away 30, I end up with a lower, I end up with hyperpolarization. Rather than being closer to my threshold, I'm farther away. So what actually happens with summation depends on the sum result of the types of ions that are entering the cell. For this one, let's say instead of allowing either positive in or out, this is a chloride ion channel, and I'm actually adding negative chloride ions. So all these are different possibilities of what can occur at the cell body. So we have this model of the cell body, and we have hundreds of neurons forming synapses. Some are allowing positive ions in the cell, some are allowing negative ions in the cell, and the net event is how many positive ions to get to the axon. So it's a little, we started out very simple, okay, but the sum total is what the event is. So here's our, I don't want stimulus yet. So in summation, here we have a cell body. There are 2 neurons that are allowing positive ions into the cell. Here is a neuron. Summation is described on the bottom of page 12. Okay. So here are some neurons that are allowing positive ions to leave or negative ions to come in. If the inside of the cell becomes more positive, that rated potential is identified as an excitatory postsynaptic potential. Not ESP, it's not extrasensory perception, but excitatory postsynaptic potential. An excitatory postsynaptic potential is a depolarizing potential. Okay? It's going to bring the current inside the cell closer to what will open the voltage-gated channels. So this is depolarizing. An inhibitory postsynaptic potential, an IPSP, is going to be a hyperpolarizer. It'll make the inside of the cell more negative. Now there are 2 types of summation. Yes?
Can you say again what you meant when you said depolarizing? That it brings it.
The inside of the cell is more positive.
It brings it closer to.
It brings it closer to the current required to open voltage-gated channels. And that word is threshold potential. We're going to talk about that next. But it just brings the inside of the cell closer to what will open our voltage-gated channels. Whereas an IPSP takes the inside of the cell farther away. It's going to take a greater number of positive ions to get to that point.
So it's increasing the threshold?
Uh-huh. Well, the threshold is still the same, but it increases what we need to reach threshold. All right, so we have 2 types of summation. We can have 2 neurons here firing nearly at the same time, and that's called spatial summation. All right, so for spatial summation, can I erase these terms right here? You guys have those down, the green and the black words? Okay. So we have spatial summation, which is 1 or more, well, 2 or more neurons firing. So spatial means in space. All right, so at two different locations on the cell body, from two different neurons, we are opening channels. It can be either an EPSP or an IPSP. Or we have temporal. And in this case, it's one neuron, which is firing multiple times. One neuron over and over. So spatial is, say, 3 neurons firing at the same time. Temporal is 1 neuron firing 3 times in quick succession.
Is there a number? 'Cause
No.
you mentioned 3, just it's kind of.
It's just, yeah,
One
it
uses-
varies on
okay.
what's stimulating it to occur. So at any rate, these are all different forms of a graded potential. Okay? And the key thing to a graded potential is it can be different strengths. And depending on its strength, depends on how long it lasts and how far it gets through the cell, whether it makes it all the way to the axon. A threshold potential is a specific type, well, is a specific event that happens because of a graded potential. So a threshold potential is a graded potential whose strength meets the minimum current required to open a voltage-gated channel. So remember the analogy of the grocery store door open because of a, say, 30 pounds, it has to be 30 pounds. So if I do an EPSP, all right, that's like putting 2 10-pound cement blocks on the mat. It's not enough to open the door, but then when a 10-pound rat runs across, the door will open. By itself, the 10-pound rat would never open that door, okay? But because of EPSPs, we raise the resting membrane potential temporarily, and now the next stimulus, even though it might not be very strong, is enough to open the voltage-gated channel. By itself, it wouldn't do that. Okay? So the threshold then is that minimum current. For the door, it would be the 30 pounds. That door is not going to open unless from some different, who cares whether it's one rat, one 30-pound rat, or 3 10-pound rats, or a child that weighed 30 pounds. The door doesn't care. It has to be 30 pounds. And then the door will open. Okay? So the threshold potential is the minimum number of ions of our graded potential that reach our axon and will cause the voltage-gated channels to open. Remember those paddles we talked about earlier? So if it needs, 10,000 positive ions to open it, and it only has 8,500, that voltage-gated channel won't open. All right? So when we diagram, may I erase this part of the board now? Okay? So when we diagram that on our graph, most of the time, and I'm just assigning a number off the top of my head, 98% of the time, our channels open because of a positive current. Rarely, and this has to do with some of our special senses, the channel is open, will close because of the current, the inside of the cell becomes more negative. All right, but we'll talk about those specific cases. In almost every other case, it's going to be a positive current. So when we look at our graph again, and our resting membrane potential is down here. The threshold, and this again is going to vary from neuron to neuron. It's given an example. Sometimes a neuron's resting is given as minus 90, but typically it's minus 70, and typically the threshold is given as a minus 55. That means that we have to add enough positive ions to bring the difference from inside the cell to outside to 55 millivolts of current. So we have to have enough, the difference here is, what, 15? Enough positive ions to create that difference. Okay? So, again, with our graded potential, if we only add in enough positive ions to get to a minus 60, nothing's going to happen. And those ions will dissipate and leave the cell, and we're back to our resting membrane potential. If we get to 50, nothing's going to happen for this particular neuron, and those ions will dissipate, and we get back to our resting membrane potential, and no action potential is created. But now let's say we have summation, and we have a whole bunch of neurons firing on this cell, and now we reach our threshold potential. Enough positive ions have entered the cell so that enough of them get to the axon hillock to reach the minimum number of ions going by to change the shape of those paddles and open up a voltage-gated channel. And that's what the threshold is. Okay? Different neurons will have different thresholds. But for a particular neuron, it will always be the same value. Unless we change the environment by changing the number of potassium ions or that type of thing. Everything else is the same. It will always take the same minimum current to open the channel to reach that threshold. Now, what happens? I'm going to go just explain really briefly the action potential, and then we'll take a break and come back and look at the variety of steps and finish up with the synapse. And then we'll leave that for get back to Tuesdays when we look at neurotransmitters specifically. So when we look at the electrical activity of what's happening at the axon, we look at this diagram. It's a very common, various forms of this diagram are out there. This is one that shows the resting membrane potential at 90. But I'll stick with the 70 we've been using.
70.
Yeah. Minus 70. So we have our resting membrane potential. And again, remember, it's the leak channels and the sodium-potassium pumps and the negatively charged proteins that are keeping it as a resting membrane potential. And then we activate neurotransmitters released, ligand-gated channels open, and we create a graded potential, which finally reaches threshold. So here is our threshold. And once we reach threshold, which is this value right here, and we're at a site where voltage-gated channels are present, they will open. We might reach threshold there at the dendrite, but there's no voltage-gated channels, so nothing happens. Okay? The current has to have access to a voltage-gated channel. When that happens, the first type of voltage-gated channel to open is a sodium voltage-gated channel. And it opens very rapidly and then closes. All right? It opens the same amount for the same time. And so it opens very rapidly and allows so many ions to enter into the cell that the cell becomes more positive inside the cell than outside. And so it'll reach a voltage of a plus 30, meaning it has the plus, meaning it has more ions inside the cell than outside. While the sodium channels are open, the potassium channel, voltage gated channel, takes its time. It's a little bit, takes longer for it to change its shape. And it doesn't actually become open until the sodium channel is closed. So the sodium channel closes, the potassium channel opens, and we start to lose positive ions. And we rapidly return even beyond the resting membrane potential because our potassium channels stay open for a longer period of time. All right? This, the graded potential is depolarization. And what kind of channels is that due to? Lightning gated channels. Probably for sodium. Most of the time it's for sodium. And then here with our action potential, so this whole event here, the blue from this point down to here, is an action potential. That's not a halo, I'm just encompassing both. Depolarization. And then, of course, we're repolarizing as we come back. And hyperpolarization. And then finally, we're back at our resting membrane potential. So what's happening in depolarization?
More positive.
Voltage gated, sodium channels open. And then close. Repolarization, voltage gated channels, potassium channels open. They're still open down here. And then they close. And sodium and potassium pumps recover the membrane. So that's why we kind of dip down a little bit. So those are the events that are listed here. So this spike is the change in current at one small, tiny, one tiny membrane area of our axon. It's going to be repeated over and over and over throughout the length of the axon. So let's stop here and take a break, and then we'll come back and look at the specifics of when and how these gates open and how we propagate this action potential along the membrane. Because if we don't propagate it, it's going to die out and look just like a graded potential. What makes it an action potential is that it is self-propagating. All right, come back at a little bit after 12. And then I'll probably lecture till 1:45 or 1, and we'll be done. These images are on page 18 of your lecture notes. So it sounds like a bit of extra detail, but what we're going to look at is this image right here. Where am I going?
You're going to be like, it's this page, and we're like, well, this one says this.
So when we look at this image, which you have on page 16, showing the action potential, you'll notice that the depolarization and repolarization before we get to hyperpolarization is indicated as the absolute refractory period. What that means is that during the absolute refractory period, if I provide another stimulus, the neuron will not react to it. no matter how high or how strong the stimulus, for the first half, it won't react because the sodium channel is already open. You can't open a door that's already open, okay? So if I say, every time I push the buzzer and the door opens, if I push the buzzer when the door opens, you're not going to see any change. The door's already going to be open, all right? Then the door closes, and during the rest of that time of the absolute refractory period, even if I push the button 1,000 times and it's not going to open. So we're going to explain why with these sodium gates. So that's called the absolute refractory period, when no matter how strong I try, no matter how strong a current I send, that sodium voltage gated channel won't open. The relative refractory period is during the recovery of hyperpolarization. And at that time, if I send a particularly strong, it has to be higher than threshold, if I send a particularly strong signal, then I can get that sodium channel to open again, all right? But only if I send a signal that's stronger than threshold. Remember, threshold is always the same? Well, threshold becomes higher during the relative refractory period. So what is it that's changing about the events that cause the sodium channels to open? And that's what we're going to go through on page 18. What is it about these channels that make them, well, it doesn't completely explain it, but it starts to explain it a little bit. All right, so back to step 1. We are at the resting membrane potential. Okay? So step 1 is, on our diagram, is over here. Resting membrane potential. And in the axon, we have voltage-gated channels for sodium. The voltage-gated channel for sodium has 2 gates, as you can see here. All right? We're at the resting membrane potential. It looks like this. One gate is closed and one gate is open. Okay. The gate that is closed is called the activation gate. The gate that is open is called the inactivation gate. The key thing is that in this particular conformation, with the activation gate closed and the inactivation gate open, this sodium voltage gated channel is sensitive to the threshold potential. This top gate, the activation gate, will open when the threshold potential is reached. Okay. So when we're at resting membrane potential, when the membrane potential is -70, the sodium, this would be the axon right here. Okay, so it's not two plasma, it's not a double layer of a plasma membrane. Clarify that. I can draw this through like this. So this entire structure here is the axon. So for this view here, you're just seeing that top one. So this is just a diagram of that top row. Okay? But it's traveling down both sides of the plasma membrane. Okay? Now I'm not going to put it in, but our potassium channel only has one gate. I'll draw that down here below. So our potassium channel only has one gate, and and it is closed during the resting membrane potential. But the key thing about the sodium, that's the one that's going to create a new threshold, if you will. The key thing about that is that it is sensitive to the lowest value. Our threshold potential was the minimum current required to open our voltage gated channel, and that's what will open the activation gate. Right now, it's closed, and during our resting membrane potential, no sodium is coming into the cell and no potassium is leaving the cell. That's why we're still at a resting membrane potential. So then we get our graded potential coming through, and the threshold potential is reached. That threshold potential is the current required to open the sodium channel and to open the potassium channel. All right? The sodium channel opens very fast. And so our sodium channel is going to look like this. So this is going to occur when threshold potential is reached and during depolarization. So this is the threshold potential being reached, and that's depolarization. We are now in absolute refractory period because our door is open. Another stimulus, another current coming down the neuron isn't going to open the door because it's open. So we're in this area right here. Again, the sodium gates are open. Potassium gates are starting to open, but they're still closed. There's no potassium going through, so we're still going to look like this. Okay? The potassium channel is still closed. But because our sodium channel gates are both open, we see a rapid depolarization as sodium ions rush into the cell. Almost as rapidly as they open, they close. And what closes first is the inactivation gate. Okay? So step 4 is at the top beginning of our repolarization and continuing repolarization. So notice what just happened. The inactivation gate closed and the potassium gate opened. We see that occurring. Okay. So now during step 4, which is here and continues down, these events, the next step that occurs is this gate stays open, but this gate closes. And our potassium channel is going to be open. So because one of these gates is closed, should we open? Because one of these gates is closed, sodium ions still can't come into the cell. Okay? So the inactivation gate inside the cell near the axon is closed. and this is still our absolute refractory period. So the reason we were in absolute refractory period here is because the door was open. During depolarization. During repolarization, we're still in absolute refractory period. Because no current strength, no matter how strong, will open that inactivation gate.
The top gate is open.
The top gate is open, the outside gate is open, but the inside gate is closed. The opposite was true during our resting membrane potential. In our resting membrane potential, the inside gate is open and the outside gate is closed. This outside gate, and this conformation, this is sensitive to the threshold current. In this conformation, it's not. So no matter how strong a current we send down here, it will not open this channel. So now we're at this period. So from here to here, actually from here to here, this neuron will not respond to another stimulus at that channel. That channel will be staying closed. That's why it's absolute refractory. No matter how hard we kick its butt, it's not going to respond. Okay? Potassium channels took a long time to open relatively. So now they're open and we're losing potassium very rapidly. If our sodium gates were still open, that would cause us to, the inside of the cell would take longer to become negative. Because we'd be adding positive ions at the same time we were losing positive ions. But since these gates have closed, we're no longer adding positive ions, we're only losing. That happens very rapidly. Okay, so resting membrane potential, sensitive to the threshold, and they open. While they're open, they're in absolute refractory period. They are insensitive to any current.
And that's in step 3 or,
This is step 2, 3, and 4.
okay.
Okay, so if we go back to this diagram about the absolute refractory period, that's occurring here. So this is step 2 right here, step 3, step 4. So now we're done with step 4 and we get to the hyperpolarization that occurs because our potassium channels stay open a little bit longer than they need to. So in step 5, we close all of our sodium gates. So our potassium gates are still open. But we're going to close this one, and this one was already closed. So this is step 5, hyperpolarization, and this is relative refractory. You would think with both gates closed that it would not be susceptible to any current, but during the relative refractory period, it is sensitive only to very strong, higher than threshold. So, super threshold stimuli. during the absolute refractory period, even a superstimulus wouldn't cause them to open. During the relative refractory period, a threshold won't, but a super will. Now, why is this important? Let's go over it one more time and talk about what's happening, and then we'll see why it's important that the cell have a refractory period. So remember, we're just talking about one tiny little piece of membrane in the axon. This is going to happen over and over and over, all the way down the axon. But we're looking at just one small fraction of a nanometer in our axon membrane. Let's go back to the beginning here. So nothing is happening to the cell. All right? The neuron is not being stimulated. And then we create the other neurons that are having a synapse with it, fire off neurotransmitters, ligand-gated channels open. We create a whole bunch of graded potentials within the cell body. Summation occurs. All of those positive ions flow towards the axon and meet up with their first voltage-gated channels. There's enough ions in that graded potential to reach the minimum threshold required to open our sodium voltage-gated channel and our potassium voltage-gated channel. This is the way it looks when that current first gets to the axon. All of our sodium-gated channels look like this, and all of our potassium-gated channels look like this. Both of them are closed, because again, remember, we're in a resting membrane potential. So in a resting membrane potential, none of these voltage-gated channels are open. But then that flood of positive ions reaches them, interacts with the negative charges in our voltage-gated channel, swings the paddles open. The sodium-gated channel opens first and looks like this. So the outward external activation gate opens, and now sodium ions rush into the cell. And that's what causes our action potential spike to form, as the sodium ions are rushing into the cell. At the same time, the current is obviously reaching our potassium channels too. But it takes them a lot longer to change shape. So while the sodium channels are open, the potassium channels are not. Yes?
Is it because they require like a higher number of?
It's more likely, I don't know for sure, but it's more likely because of other interactions between amino acids, between the atoms of other molecules, that I don't know if they have to form disulfide bonds or different things have to happen for them to change shape. So it's not that they require more ions, the same current is the threshold for each one, but there's hindrances to the shape that they take that takes longer for that to happen. But it's the same current strength for each one. The threshold is going to be the same for each one. It's just internally in the protein, it takes longer for that channel to open, that shape to change. All right, so we begin the absolute refractory period. At this time, if we were to send another signal, nothing would happen. The channel is currently responding to the one at hand. All right, then we reach the peak of our action potential. By now, our potassium channel is open, and our sodium channel closes. The first gate to close, however, is the one that was open while it was resting. That's the internal gate that was inside the cell. So that closes. And we're at this point here. So this gate closes. Doesn't matter that the other gate is open. We stop any sodium ions from going into the cell. So we no longer are going any higher with our peak. The cell's not getting any more positive because this gate has closed. Right? We're still in absolute refractory period. Even though the gate is closed, we can't open it. Nothing, no current is strong enough to open it. It's not in a position where it's going to respond to any additional positive ions. However, our potassium channel is finally open. And now positive ions leave the cell. Right? As those positive ions leave the cell, that change causes the other gate on the sodium channel to close. And at that particular current strength, and in this particular position, it now will respond to a stimulus. But that stimulus has to be stronger than the one that would open it when it was in this position. Okay? That's why it's relative. So a threshold wouldn't work. It has to be super threshold for it to open. Finally, and our potassium channel is still open. So we're at this point here. Finally, the potassium channel closes, sodium potassium pumps redistribute the ions on either side of the membrane where they're supposed to be, and the inactivation gate opens into the cytoplasm, and the cell is now ready to respond to our normal threshold potential. So why is that important? I've been talking as if when our threshold potential, when our graded potential entered the axon, that it was only going in one direction. I kind of drew it. When I drew the cell, remember how I showed ions going out into the dendrites? Okay, so, may I erase this diagram here? Okay. When we open our sodium gated channel, and I'm not going to go through all these fancy diagrams here, but here's our axon. When we open this channel, okay, I won't go through that. It's closed at first, right? And then the current reaches it, and it opens. More ions come in. Sufficient number of ions that they reach the threshold of the next channel. So the next channel is going to be able to open. Well, the current's going to go this direction, but the current's also going to go this direction. There's nothing that says the diffusion only goes in one direction. The ions are going to diffuse in both directions, right? I mean, we're assuming, because our action potential goes away from the cell, that that's the way the current goes, and it is. But the ions diffuse in both directions. So why don't we have an action potential forming in the cell body? Exactly. So when these ions flow back, let me do this a little bit different here. When these ions flow into the cell body, there's no voltage gated channels. So we don't make an action potential because there's no voltage gated channels to respond. These have ligand gated channels. So even though the ions go that direction, we don't see any result. All right, so it drives it down this direction. So it opens this channel. Now these channels are starting to recover. This channel is not going to respond because it's an absolute refractory. It's already open. So even though the ions flow by it and it's a pretty strong current, who cares? It's already open. There's nothing to do. This channel has now closed. And should probably draw this a little bit bigger. It's in relative refractory period. What's happened to our current? So as this current comes in, it goes that way, and it goes that way, it's going to get weaker. Because it couldn't open that channel to regenerate a new current. By the time it gets to the next channel, it's a little bit weaker, and it has to be stronger to be open or relative refractory. But instead it's gotten weaker. So it's not strong enough to open that relative refractory sodium channel. By the time it gets one that's recovered, our current strength is subthreshold. So it won't open this one. It has to have a strong stimulus to open this one. A normal stimulus will open this one. But what's happening is it goes this direction, it's getting weaker and weaker. So it's not strong enough to open the relative, and when it gets to the threshold, it's less than threshold, so it won't open that one. So that's why we see the effect is only downstream. That's the importance of having absolute and relative refractory periods, because otherwise we'd be setting an actual potential in both directions. And reconfusing which way, when does it stop going both ways and crisscrossing over itself. So the presence of absolute and relative refractory periods ensures that we send the signal in one direction. Okay. Yes?
Does each voltage gated channel down the axon require like a higher
No.
current?
They all require the same amount, and so that's the purpose. This is really just like a graded potential. If this had no more voltage gated channels, this would die out, and that would be the end of our passage. That's what happens with MS. We lose, we'll talk about the myelin sheath in just a second. We lose the myelin sheath, we create this voltage gated channel, it creates a current, and then peters out. So let's look at what happens with the myelin sheath. So this is sometimes called saltatory conduction, when we have a myelin sheath. And that's really because the current doesn't really jump. This is just on the bottom of page 20. You can just add it to that. So remember that myelination keeps the ions from leaking out, all right, being pumped out as we recover. Normally we would come back to our resting membrane potential. So at the node, the axolemma has access to extracellular fluid where there's sodium ions and potassium ions, and we have the presence of voltage gated and sodium and potassium channels. So what we do is, instead of having to regenerate the current every little fraction of a millimeter, with the myelin sheath, the current can now, it's not going to leak out, it's not going to get weak, and so it can now travel all the way down here and then get regenerated and travel the next distance. So let's say that I have, I'm having a race with my sister and her husband and our kids. So each of the families has a kid that's volunteered to sit on a wide skateboard, maybe a longboard, okay? And I'm going to push my kid, and my sister's going to push her kid. And if it was just the two of us, eventually, because of resistance, friction, the skateboard would stop rolling, okay? So how could I keep the person that's on that skateboard moving? Or what if I'm going uphill? How could I keep them moving?
Keep
More
pushing them.
people pushing.
I could push them through my husband. So I push, they roll, they get to my husband while they're still rolling, and he pushes them again. And then another person pushes them again. So that's what's happening at each of the nodes of Ranvier. The current is created, it flows through, and before it can completely die down, and it's kept from dying down because of the myelin sheath. So maybe, we've got them on a frozen lake and their skateboards on ice skates, or we've oiled or whatever. So we've decreased the resistance. So our current travels a longer distance, and then we renew it at the node of Ranvier by sodium channels, voltage-gated sodium potassium channels. So at every node, we renew that current instead of right next to itself. And that takes a decreased amount of time, rather than having to open channel after channel after channel. We can send that current flowing through the cell, renew it, send it flowing through the cell, and that occurs at a much faster rate. So the current appears to jump from one node to the next when actually it's hidden underneath here and traveling through the cell. Okay. So this brings us to a comparison of speed. Just on general purposes, you already knew that a myelinated neuron, the current traveled faster. But size is also a factor. If a cell is in large diameter, it has more plasma membrane and creates more current, it's going to travel faster. Okay? So if it's large and myelinated, that's the fastest traveling action potential. If it's small and unmyelinated, that's the slowest traveling action potential. So we seem to see the fastest with skeletal motor. So if I decide to turn my head or lift my leg up, that's going to be traveling on large diameter myelinated axons. If I'm trying to figure out if I'm touching my face, my daughter's face, or my son's face, proprioception, apple or orange, peach or baseball, that's also going to travel on large myelinated neurons. For some reason, pain's not that important. Pain is myelinated, but it's kind of an intermediate diameter. Even less important are autonomic motor fibers, so regulating my heart rate, which you would think would be important, regulating the diameter of my blood vessels or my airways. Okay. Those are set on small diameter fibers that aren't myelinated. Now these are given letters. In some classifications, they're given numbers. Okay, so 1, 2, 3, 3A, 3B. And I don't know what determines, how the body determines how important which modality gets what speed. Okay, do you want to take a short break, or you want to keep pushing through for 15 more minutes and be done?
Let's pile through and get done.
Okay. I figured that's what you would say, but I decided to make it your decision rather than mine, and maybe you won't trouble. So we're going to finish up by taking the action potential, taking this current all the way to the end of the neuron, and see what happens when it reaches the synaptic bouton, or the synaptic bulb, which is actually where we started, by looking at the interaction between the synaptic bulb and our cell body. So now we sent this signal down the end of our axon, and it's reached the axon terminal, and it's reached the synaptic bulb. So what happens once it gets there? You're still taking down this information. May I erase the board?
Wait, one.
Okay. variety of action potential animations that you can find on YouTube. If you have My AMP, there are some on there as well. Again, with the old adage, 1,000 pictures worth 1,000 words, especially when it's animated. All right, you can stop and start it. Instead of my drawing these ions going through the channels like I showed you yesterday, you can see that happening. And so if this seems confusing, before you go back to study it from your notes, try to watch one of these videos. And I think you'll make your notes a bit clearer. I'm animated, but I'm not dancing. All right, so a synapse is the interaction between two neurons. When we finish this unit and we talk about muscles, we'll talk about a neuromuscular junction, which is the interaction between a nerve and a muscle. Okay? And when we talk about two neurons, we're talking about a synapse. And there's a variety. There's dendrite to dendrite, and there's axon to dendrite, the most common. There's axon to cell body. Occasionally there's axon to axon. But we're just going to focus on the dendrite to cell body, the axon to cell body. A very few synapses are electrical synapses. Remember when we talked about gap junctions? We're talking about tight junctions and desmosomes, and gap junctions were part of the conversation. So between one neuron and another neuron, in a few locations, rather than having a chemical release and opening an ion channel, the two cells are connected by two ion channels, which are the gap junctions. So there's no chemical involved. The chemicals of the current flow from one cell through the gap junction to the next cell. Most of the incidences of this occurring between neurons, we often have it in muscle cells, but most of the incidences of this occurring in neurons is in embryonic development. We do occasionally that some of the neurons in the hippocampus in memory have electrical gap junctions. Part of the reticular system does, but not very often. There's not very many. By far, the most common type of a synapse is a chemical synapse, where a cell is releasing chemicals. So when we look at what is happening, again, we have our axon terminal and a synaptic bulb, or synaptic gluton. And we have voltage-gated sodium and potassium channels. And our action potential continues to form and reform like dominoes along the plasma membrane, the axolemma of our axon terminal. And that sends the signal, the electrical current, down the membrane to the synaptic gluton. That's our impulse. And when it reaches the action gluton, the same thing happens. We have sodium and potassium ligand, potassium voltage-gated channels. However, we add an additional voltage-gated channel, and that is for calcium. So in this membrane, we have a calcium voltage-gated channel that is opened by the current created when the sodium voltage-gated channel opens. So I have another sodium, it's a calcium. When the current reaches our synaptic gluton, calcium ions enter the cell. Calcium ions are often used as a signaling effect, and they activate a sequence of events inside the synaptic gluton. We have synaptic vesicles, and these are the storage sites for our neurotransmitters. So we've been using acetylcholine. Let's continue with that. So this sac has acetylcholine. And we only want this sac to fuse and activate exocytosis at certain times. We don't want that happening just randomly. Having the membrane sac bump into our membrane and dump all the neurotransmitters out, and then things fire when you don't want them to fire. Okay? So that's a controlled activity. There is a protein within this membrane that allows it to interact with the plasma membrane called synaptotagmin. Synaptotagmin isn't working and will not allow our vesicle to bind with the membrane unless calcium interacts with it. So as soon as calcium interacts with the synaptotagmin, when this vesicle bumps into the membrane, it will fuse with it. Okay? So now this can as long as it has these activated synaptotagmin molecules, be able to release the neurotransmitter. I want to say a little bit about lab, but that's the end of lecture. So on Tuesday, remember I said as important as the action potential is, it's the neurotransmitters and their receptors that give us the control over once that signal is sent, it's gone. Okay? So what we do is we control when and how often that signal is sent. And so that depends on the neurotransmitters and their receptors and whether we block them or activate them. So your lab activity is, for the first time, we're using the PhysioX book. This should have come in your packet when you bought the textbook. If you didn't, it should be in the My AMP if you decided to buy that separately. My AMP Access came with your textbook if it was new, so you can use the DVD in here or you can use the PhysioX online. If you don't have either, you can buy somebody a chocolate eclair or bribe them somehow to let you work with their DVD or use their access. There are 2 labs in here, the muscle and the neurophysiology one that we're doing, and then about 3 or 4 that we do next semester. Okay? So that may influence whether or not you decide to beg, borrow, or bribe, or get your own. In your lab packet, I don't know if you have it with you, but in your lab packet, I'm thinking it's lab 24, 25. Should be the first one that far back. 30. What lab here?
Number 30.
Okay, so page 7 or page 9. If you look at page 7, it says PhysioX 8.0. That's actually my favorite version. Now what they print is PhysioX 9.0. Okay? If you have PhysioX 8.0, you're going to use page 7 and 8 because the questions and the activities are slightly different. If you have PhysioX 9.0, you're going to ignore pages 7 and 8 and you're going to start with page 9. Does that make sense? Okay. The difference in PhysioX 9.0 and the reason it's actually better as a learning tool, but it becomes very frustrating. One of the differences, they don't have the same experiments, and I missed one that they do have. I really think it was important and they left it out. But what's frustrating for me is each time, so what happens is you go online or you use your DVD And you read a little bit about neurophysiology and what we've been talking about the last 2 days. And then it gives you a screen. These are simulated nerve experiments. It gives you a screen with, say, a nerve cut out of a frog, a sciatic nerve. And these are actual labs that students do at Sac State for their upper division physiology class. Or you can take a nerve cell and work with it. All right, so on these screens, let's get to it. Unfortunately, they do skeletal muscle first when they haven't figured out how the nerve impulse is made. So the full screen on the computer will be an oscilloscope, kind of like an ECG. You see a little bead of light, and the nerve preparation. And so you're going to, in the past, add a cold bar to it, or drip acid on it, or drip something warm or something cold and see how that changes it. You might put an electrode inside the nerve and turn up the current and see what the threshold potential is before the axon fires. So there's a different series. You can put drugs, different drugs on it. Use a rat neuron and different diameters of neurons to see how fast the current travels. So it's a different series of experiments, and then it asks you questions. You don't have to turn anything in from this book. I have written the questions I want answered in your lab manual. I've taken them from the book, and the tables that I want the information for, I have already typed out. All right? So some of the tables will have the voltages already there, and you put down how the voltage changes. So those are already, so what you're handing in is what's in your lab packet. Now the frustrating part, for me anyway, it's a good learning tool, but before you get to do the experiment, it will ask you questions. And then you answer them. For some faculty, those answers are sent to them by email, and it affects the students' grades. What I'm grading is in your packet already, so that's just something for you to answer, and you can guess, or you can figure it out. It's up to you. You're going to learn more by trying to figure it out. But if you go back to repeat the experiment, you have to answer the questions all over again. So every time you try to do it, you can never just opt to not answer the questions. So as a faculty member trying to write up the lab and going over the experiments multiple times, I had to sit there and just put in an answer somewhere and move on and just try to get to the experiment. So that's why I said it was frustrating, because you can't just opt out once you've done them, answer those a few times. Okay? But if you're doing it online, it helps to have this book, because you can read the directions and have the picture and the experiment online. If you're doing it completely online, open 2 different windows so you can toggle back and forth between the directions and the actual experiment. A lot of the directions will be on the side. It'll say do this next or do this next. Okay, but it's still helpful to have another window if you're not using the book to tell you what to do next. So this is a simulated wet lab. You don't have to wear goggles while you're doing this, unless you want to. Just pretend that you're actually working with a nerve. Closed-toe shoes and all of that. All right? But as you go through, and I'm not having you do every single experiment, so if you look at your lab at the beginning, where I give the assignments, it will tell you which ones to do. And it'll say do exercise 1 and 2 and 5 and 6, 7 and 8 type of thing. So read that first. And you're welcome to do all of them, but the ones I want you to answer questions about are going to be listed on the what to do kind of portion of the assignment. Okay? So this is your first work with the exercise viz thing. Some students like it, a lot don't, because it's kind of a cookie cutter method. There are some out there where they're randomized and everybody gets different answers because they have different values to start with, but it's a good starting point to get you in understanding what's happening. I would still watch a video of the action potential before you start the lab, just to give you a visual of what's going on with the channels. Alrighty?
I have a question. With the slides that you used today, are those actually the lecture slides, or is this something you put together?
They're the same packet as yesterday's. So you have them because you copied. I still need to double check to see that they're actually on D2L.
Okay.
But yeah, these are,
Cool. Okay.
I didn't do anything new for today,
Okay.
if that was out of your asking.
Yes. Alrighty.